Hello, my name is Godfrey, and my abstract project is on the quantification of transcripts of intrarenal renin-angiotensin system in male and female mice and rats. So for a little background on my project, the intrarenal angiotensin system, or RAS, is responsible for regulating the body's blood pressure and electrolyte balance. When RAS is inappropriately activated, hypertensive injury, kidney injury can arise. Studies suggest that females are more resistant to this injury compared to their male counterparts. Mice especially seem to have a lower sensitivity to kidney injury compared to rats. However, the molecular mechanisms of this have not been well elucidated. The question of this project then was determining whether intrarenal RAS contributes to the pathological differences seen between sexes and species in hypertension and kidney injury. The goal of this project was to determine absolute copy numbers of intrarenal RAS components in male and female mice and rats using digital droplet PCR, or ddPCR. So we began the experiment by first harvesting the renal cortices of 6 hypernormotensive male and female mice and 3 male and female normotensive rats. The RNA was quantified with nanodrop. For absolute quantification of genes, ddPCR technique was used. The project focused on 6 key RAS components. These components were renin, angiotensinogen, AGT, angiotensin converting enzyme, ACE, ACE2, angiotensin 2 type 1 receptor, AT1R, and AT2R. So here are the results for the experiment. When looking at these graphs, an asterisk above a bar indicates that the difference between the comparisons was statistically significant, and the y-axis in this data signifies the concentration of copy numbers in 1 nanogram of RNA. So for instance, with the male-female comparison of our mouse samples, there was a statistically significant difference in the AT2R and ACE. And in AT2R, females were increased, while in ACE, females were statistically significantly decreased compared to their male counterparts. And I would like to take a moment to point out here that across all samples, AT2R was found in much lower concentrations than any other RAS component. And this finding is actually supported by other studies that found similar findings. With rat comparison between male and female, as you can see, AGT and AT2R had significant differences, with females having lower AGT than males and females having higher AT2R than males. When comparing male RAS between mice and rats, we found that renin, AT2R, and ACE had differences, with renin and ACE both being markedly lower in rats, and AT2R being markedly increased in rats. Finally, we have the female intrarenal RAS comparison between mice and rats, and these were similar findings that the males with lowered levels of AGT, renin, and ACE. So what do these numbers tell us? These findings suggest that intrarenal RAS component expression is dependent on both sex and species. These differences could account for the different pathological severities under hypertensive conditions. Female resistance to hypertension and kidney injury, for instance, may be supported by these lower levels of AGT and ACE seen in the study. Moreover, the lower sensitivity and angiotensin 2 induced intrarenal RAS activation and kidney injury of mice might be supported by this greater baseline expression of renin and ACE that is seen. So I would just like to thank everyone that helped me with this project and my mentor in the Department of Cell and Molecular Biology, Dr. Robert Dotson, that oversaw this project. And thank you all for your consideration.